So, hi everyone. Today is the 12th problem solving session for the course Environmental Chemistry and Microbiology. So, today is the last topic of this course that we'll discuss. We'll discuss about biogeochemical cycles today and some other metabolic diversities. The course instructors, as you know, are Professor Anjali Pal and Professor Sudha Goel from IIT Kharagpur. I'm Shrija Ghosh of PMRF from IIT Kanpur. And today I'll be solving some questions related to your 12th week's assignment, and feel free to ask any questions that you have regarding your assignment and regarding any of the topics in this session or regarding any of the questions that I am discussing. Just if you have any question, let me know. Okay, we'll start with the questions now. Starting with the first question. Consider the following with respect to carbon cycle: ocean, forest, soil, and abandoned mines. Which of the above mentioned cannot act as carbon sink? So you have ocean, forest, soils, and abandoned mines. Which of the above cannot act as a carbon sink? So option A is first and second only, that is ocean and forest only. Option B is 1, 2, 3, and 4, so all four. Option C is 3 and 4, that is soils and abandoned mines. And option D is fourth only, that is abandoned mines only. So which of the above cannot be considered as carbon sink? Anybody? Whoever knows the answer, you can write the answer in the chat box and then we can have a discussion. So which of the above mentioned cannot act as a carbon sink? Ocean, forest, soils, and abandoned mines. Write your answers in the chat box. Even if you don't know the answer, try to guess the answer and then we can have a discussion if you are right or wrong. Okay, I have one answer in the chat box. That is option D. Okay. Anyone else wants to answer? Okay, let's see if option D is correct or not. That is the correct answer. So only abandoned mines cannot act as carbon sink. All the rest can be considered as carbon sink. So a carbon sink can absorb more carbon from the atmosphere than it would release. So for example, plants, ocean, soil. So you had ocean, forest, and soil. So all of these can absorb carbon from the environment more, like they can absorb more than the amount they can release. Whereas mines, as you know, they are a source of carbon. So they are uh, like they are not carbon sink, they are not absorbing carbon, they are the source of carbon. They can provide carbon, they can release carbon. Okay, so that is a carbon source. So you have two terms, carbon sink and carbon source. So carbon sink basically something that can absorb more carbon from the atmosphere than it would release, and carbon source would release more carbon into the atmosphere than it can absorb. So examples for carbon source are volcanic eruptions, burning fossil fuels. Even mines are an example of carbon source. Okay, so among the given options you have abandoned mines that cannot act as carbon sink. Okay, so two people have answered option D, so both of you were right. Okay. Anybody has any doubt in this? Oh, great. Then I can move to the next question, question number 2. What organism represents the symbiotic relationship between the roots of a plant and fungi? What organism represents the symbiotic relationship between the roots of a plant and fungi? Option A is legume, option B is mycorrhiza, option C black root, and option D lichen. Which organism represents the symbiotic relationship between the roots of a plant and fungi? Legume, mycorrhiza, black root, or lichen? What is the correct answer to this? Write your answers in the chat box. All of you write the answers in the chat box. I have one answer here, option A. What about others? What organism represents the symbiotic relationship between the roots of a plant and fungi? Option A is legume, option B is mycorrhiza, option C is black root, and option D is lichen. Okay, I have another answer in the chat box. Says option B, that is mycorrhiza. Anyone else wants to answer? Anybody? Okay, let's see the correct answer. The correct answer is option B, mycorrhiza. So mycorrhiza is the symbiotic relationship between the roots of a plant and fungus. So it is generally so it's a fungus. So basically, it lives in close proximity with the roots of the plant. And a symbiotic relationship means both the organisms are benefiting from that relationship. Okay. So the fungus and the plant is in a symbiotic relationship. That means both the plant is having some benefit from the fungus, and the fungus is having some benefits from the plant. Okay, so how are they benefiting from each other? The fungus, since it is living in the close proximity to the plant, it is living near the roots of the plant, it is easily getting the carbon source from the plant, that is the carbohydrate. So it uses the simple carbohydrates and inorganic nutrients from the soil. So it can get those carbohydrates directly, because plants, they undergo photosynthesis, right? They make their own food, they make their own carbohydrates, and that those carbohydrate, those carbon source, it can be easily provided to the fungus. So fungus is getting food from the plant, you can say. And in return, what the fungus is doing, fungus, since it is present near the root surface, and fungus is generally, so mycorrhiza is generally a mycelial fungus. So what do you mean by mycelia? You have seen like thread like fibers are there for mycorrhiza. Okay, so because of those thread like fibers, it basically increases the root surface area. Okay, roots are also thread like, and then over the roots, if some thread like fungus is growing, it is basically increasing the surface area of the root and basically increasing the absorptive area of the plant root system. So roots of the plant are basically there to absorb all the nutrients from the soil, right? So basically the fungus is helping the plant absorbing the nutrients more and more from the soil, and the plant is helping the fungus by providing simple carbohydrate. Okay, so it's a symbiotic relationship. Both the parties are benefiting from this relationship. Okay, so obligate symbionts, improve plant's ability to absorb nutrients from the environment. And the correlation between the plant diversity and abundance and the diversity of mycorrhiza, there's a direct correlation between both plants diversity and diversity of the fungus. Okay, so mycorrhiza, it is the symbiotic relationship between the plant roots and a fungus. Okay, mycorrhiza is a fungus that grows on the roots of the plant. That's clear? Anybody has any doubt in this? If you have any questions regarding this topic, let me know. Okay, moving on to the next question then. Question number 3. Oxidation of ammonia into nitrites is carried out by. Oxidation of ammonia into nitrites is carried out by. Option A, Nitrobacter. Option B, Enterobacter. Option C, Nitrosomonas. And option D, Rhizobium. Oxidation of ammonia into nitrites is carried out by. Option A, Nitrobacter. Option B, Enterobacter. Option C, Nitrosomonas. And option D, Rhizobium. What do you think is the correct answer? Write your answers in the chat box. Everybody write the answers in the chat box. I have discussed this in previous session also. The process of oxidation of ammonia into nitrites. So ammonia is oxidized into nitrites and then nitrate. We have talked about Vinogradsky column, remember? So we have 4 options: Nitrobacter, Enterobacter, Nitrosomonas, and Rhizobium. What do you think is the correct answer? I have one answer in the chat box, option C, that is Nitrosomonas. Anyone else? Anyone else wants to answer? Come on, everyone, try to answer. Okay, I have another answer in the chat box. That is also option C. So let's see what's the correct answer. The correct answer is option C, Nitrosomonas. So both of you are correct. So basically we have 4 options here: Nitrobacter, Enterobacter, Nitrosomonas, and Rhizobium. So all 4 of them have specific functions in the ecosystem or the environment. So Nitrobacter is a bacteria that helps oxidizing nitrites into nitrates. Okay, so in the nitrogen cycles, the nitrogen is fixed into ammonia, and then ammonia is then converted into nitrite and nitrate. And then later again denitrification happens where nitrates are again converted back to nitrogen. Okay, so that is how nitrogen cycle goes on. So the oxidation of nitrites into nitrate is carried out by Nitrobacter, whereas the oxidation of ammonia into nitrite is carried out by Nitrosomonas. Okay, The other two options we had Enterobacter and Rhizobium. So Enterobacter belongs to coliform groups of the bacteria, and they are mostly pathogenic and commonly cause infections in the urinary and respiratory tract. So it's a pathogenic bacteria. It generally causes UTIs and respiratory tract infections. Okay. Nitrosomonas, they use ammonia as the source of energy, and in the presence of oxygen, utilizes carbon dioxide as a carbon source. Other bacteria that oxidizes ammonia are Nitrosococcus and Nitrosospira. Okay, so three bacteria that can convert ammonia into nitrite. Okay, so this process is nitrification. Then you have rhizobium. We had another option called rhizobium. Rhizobium is basically gram-negative bacteria that can fix nitrogen. So I told you the atmospheric nitrogen is fixed into ammonia, and then that ammonia is converted into nitrite and nitrate, and then later nitrate is again converted back to nitrogen that goes into the atmosphere. So that's how nitrogen cycle goes. So that nitrogen fixation, the conversion of nitrogen into ammonia, that is carried out by these rhizobiums. So these rhizobiums are present in the roots of the legumes. So you have leguminous plants. All these uh, example of leguminous plant is basically the pulses that you have. Okay, so the roots of those plants, they have small bulb-like structure, and inside those bulb-like structure, these rhizobium, these bacteria are present, and they basically, again, they are in a symbiotic relationship. I explained you what symbiotic relationship is, right? So these bacteria also helping the plants to absorb nutrient, and they are helping the plants by fixing the nitrogen. So they are fixing the nitrogen, converting into ammonia. So the plants are getting ammonia as a nitrogen source. Okay. In return, what they are getting a place to live. They are getting nutrients from the plants. They are getting carbohydrates from the plants. Okay. So it's a symbiotic relationship between the plant root and the bacteria. Okay. So this is similar to mycorrhiza. Mycorrhiza was a mycelial fungus. This is a gram-negative soil bacteria. Okay. So this is how nitrogen cycle works. So first you have nitrogen fixation that is given here. So you have nitrogen fixation. So you can have different types of nitrogen fixation: aerobic, anaerobic, symbiotic. So aerobic, we have cyanobacteria and azotobacter, which can do the aerobic nitrogen fixation. So basically, nitrogen is converted into ammonia. Okay, that is the nitrogen fixation. Then anaerobic, we have Clostridium and Methanobacterium, which can do this process anaerobically without the presence of oxygen. Then symbiotic relationships, I've explained that they generally live in proximity with some other organism, and both the organisms are benefited by that relationship. So rhizobiums, they live in the roots of the legumes. So both the leguminous plant and the bacteria are benefiting from each other. Ammonification is basically like in nitrogen fixation, atmospheric nitrogen is converted into ammonia. Ammonification, organic waste matter and everything, these are converted into ammonia. Okay. So ammonification is carried out by Bacillus, Pseudomonas, Clostridium, and Streptomyces. Then this ammonia basically is converted into nitrite. So ammonium is converted into nitrite, and then nitrite is converted into nitrate. So this process is nitrification. Okay. So nitrogen fixation is happening. So N2 is becoming NH3, then NH3 basically ammonia or ammonium ion. It can take one proton to become ammonium ion. That ammonium ion can get converted into nitrite, and then nitrite can get converted into nitrate. So this whole process, the two-step process, this is called nitrification. So the first step of nitrification is carried out by Nitrosomonas or Nitrosopumilus. The second step is carried out by Nitrobacters. And then this nitrate is again converted back to nitrogen. That is the denitrification process. And the organisms that can perform the denitrification are bacillus, pseudomonas, and paracoccus. Okay. And you have another process called anammox. So anammox is a process which is basically anaerobic ammonium oxidation. Okay, so that is the uh, origin of anammox, anaerobic ammonium oxidation. So ammonia is basically anaerobically converted into nitrogen. So this is again an opposite process of nitrogen fixation, and this is carried out by Brocardia. Okay, so anammox is a process that is anaerobic ammonia oxidation, and ammonia is oxidized back to nitrogen, atmospheric nitrogen here, by the organism Brocardia. Okay, so these are all the processes involved in nitrogen cycle. I hope this is clear to everybody. Is this clear to everybody? If you have any doubt in any of the process or any of the things that I've just now explained, please let me know. Anybody has any doubt? No. I can move on to the next question then, if there's no doubt. Question number 4. What organism is the result of a symbiotic relationship between a fungus and an algae? So again, a symbiotic relationship, but this time it is between a fungus and an algae. Option A, lichen. Option B, mycorrhiza. Option C, legume. Option D, rhizobium. What do you think is the correct answer? Lichen, mycorrhiza, legume, and rhizobium. A symbiotic relationship between fungus and algae. I have one answer in the chat box, rhizobium. Anybody else? A symbiotic relationship between fungus and algae. Okay, both the answers in the chat box are option D. I just now explained rhizobium. Did you not understand what rhizobium is? What do you think a rhizobium is? If you are saying rhizobium is the correct answer to this, so it has to be a fungus or an algae. What do you think it is? Is it a fungus or an alga? If you are answering option D to this question, then you are agreeing that rhizobium is either a fungus or an algae. Come on, tell me. I just explained what rhizobium is. How can you answer rhizobium to this question? What is a rhizobium? If anybody heard listened to my explanation and if anybody remembers. What's the point of having these doubt clearing session if you are not going to clarify your doubts? If you are understanding everything, then let me know. Clearly, you are not understanding anything because I've just explained what rhizobium is. Tell me what rhizobium is. Come on, Ipsmita and Tumma Lakshman Rao. Both of you have answered option D. Tell me what is a rhizobium? I told you rhizobium is a bacteria that lives uh, near the roots, lives in the roots of legumes. So they have a symbiotic relationship with the leguminous plants. I explained here, right? Rhizobium is a gram-negative soil bacteria that can fix nitrogen. These kind of bacteria are known to form symbiotic relationships between roots of the legumes, enabling nitrogen fixation, right? So here in this question, a symbiotic relationship between fungus and alga, the correct answer is lichen. Option A, lichen. So mycorrhiza, I told you, it's a symbiotic relationship between fungus and roots of the plant. Legume is a plant basically. It's not uh it's a it's a plant. Rhizobium, it's again a soil bacteria and it is in a symbiotic relationship with the legumes. Okay, so legume and rhizobium are in symbiotic relationship. Mycorrhiza is in a symbiotic relationship with the roots of the plant. So mycorrhiza is fungus. Lichen is the symbiotic relationship between the fungus and an alga. Okay, so it's a symbiotic relationship between fungus and algae. Mycorrhiza is a symbiotic relationship between plant roots and fungi, and rhizobium are soil microbes that form association with the roots of the leguminous plants, forming root nodules. I told you they form small bulb-like structures on the roots, inside which these bacteria are living, and they enable nitrogen fixation. Is that clear to everybody? I hope everybody understood rhizobium, mycorrhiza, and lichen. If you have any doubt, let me know, please. Anybody has any doubt? That's clear to everybody? Okay. Moving on to the next question, question number 5. Which of the following is correct reaction representing the denitrification process? Again, I've just told you all the nitrogen cycle processes. So which of the following reaction represents denitrification process? Option A, ammonium to nitrite. Option B, nitrite to nitrate. Option C, nitrate to nitrogen. And option D, nitrogen to ammonia. Which of the following represents the correct relationship, correct reaction representing denitrification process? Ammonia to nitrite, nitrite to nitrate, nitrate to nitrogen, or nitrogen to ammonia? I have one answer in the chat box, option D. I've just now explained all the nitrogen cycle processes. All of you should be able to answer this. Write the correct answers in the chat box. What about others? Write the answers in the chat box. Hmm. So the correct answer to this question is option C, nitrate to nitrogen. So ammonium to nitrite and nitrite to nitrate, these two are the process, part of the process called nitrification. Okay. Nitrate to nitrogen is the process of denitrification, where nitrates are reduced back to nitrogen. So nitrogen to ammonia, this is a process of nitrogen fixation. Okay. So nitrogen is fixed, basically atmospheric nitrogen, it is in gaseous form, it is fixed into ammonia. So it is basically oxidized form here, and then sorry, it is the reduced form here, and then after that oxidation of ammonium into nitrite and then nitrite to nitrate. This is oxidation process, that is nitrification. Then you have denitrification, where nitrate is again converted back to nitrogen. Okay. So denitrification is again a reducing process. This is fixation, this is reduction, and these two are oxidation. Is that clear? So I had shown you here the processes. So you have nitrification, ammonium to nitrite and nitrite to nitrate, then denitrification, nitrate to nitrogen, then nitrogen fixation, that is nitrogen to ammonia, and we had anamox, where ammonia is converted back to nitrogen. Okay, moving on to the next question now. Question number 6. Under anoxic conditions, green and purple sulfur bacteria can utilize dash as a source of energy. Under anoxic conditions, green and purple sulfur bacteria can utilize dash as the source of energy. Option A, nitrogen. Option B, oxygen. Option C, carbon dioxide. And option D, light. So under anoxic condition, green and purple sulfur bacteria can utilize dash as the source of energy. Nitrogen, oxygen, carbon dioxide and light. Write your answers in the chat box. I have one answer in the chat box, option D, that is light. Okay. What about others? Everybody write your answers. I have only one answer in the chat box. Everybody else write your answers. What would be the source of energy for green and purple sulfur bacteria under anoxic condition? Okay, let's see the correct answer. Correct answer is option D, so you are right. So green and purple sulfur bacteria utilize light to reduce carbon dioxide to carbohydrates using H2S instead of water. So since it is under anoxic condition, so H2O is not available. So instead of H2O, they use H2S since they are sulfur bacteria, green and purple sulfur bacteria, right? And they are photosynthetic, so they use light to reduce the carbon dioxide to carbohydrates. So I told you in the previous class photoautotrophs and chemoautotrophs. So these are photoautotrophs basically. They are using carbon dioxide as the carbon source. So they are autotrophs and they are using light as the energy source. So they are photoautotrophs. Okay. I hope that's clear. Moving on to the next question. During anaerobic decomposition, complex polymers are broken into simple monomers in which of the following steps? Option A, methanogenesis. Option B, acetogenesis. Option C, fermentation. And option D, hydrolysis. During anaerobic decomposition, complex polymers are broken into simple monomers in which of the following steps? Option A, methanogenesis. Option B, acetogenesis. Option C, fermentation. And option D, hydrolysis. What would be the correct answer? Write your answers in the chat box. Come on, write your answers in the chat box. How will the complex polymers, how will be the complex polymers broken down into simple monomers? So breakdown of polymers, what would be the reaction? Methanogenesis, acetogenesis, fermentation, or hydrolysis? Everybody write your answers in the chat box. Even if you don't know the answer, at least try to guess the answer so that you will have some idea. Think logically and answer. Anybody? Nobody knows the answer to this? Breakdown of polymers into monomers. So the correct answer is hydrolysis. So all the complex polymers like carbohydrates, protein, fats, they are broken down into their monomers. So carbohydrates are broken down into sugars, proteins are broken down into amino acids, and fats are broken down into uh, fatty acids. Okay, so all these polymers are then broken down into monomers. So this breakdown process is basically called hydrolysis, and these are carried out by some cellulolytic or hydrolytic bacteria. After hydrolysis process comes the fermentation process. That was the one of the options, the third option. So monomers, the sugars, amino acids, and fatty acids, they undergo for different type of fermentation process. So sugars are generally broken down into carbon dioxide and hydrogen. Uh, then some fatty acids, they are converted into acetates. And then we have some sugars are also converted into propionate and butyrate. Okay, amino acids are also processed like that. Then comes acetogenesis, which was again one of the options. So carbon dioxide and hydrogen, these are converted into acetate by the process of acetogenesis. And once we have acetate, and propionate and butyrate is also converted into acetate. And once we have the acetate, then methanogens convert the acetate into methane and carbon dioxide by the process called methanogenesis. Okay. So you have methanogenesis, acetogenesis, fermentation, and hydrolysis. So first step is hydrolysis, then comes fermentation, then comes acetogenesis, and then finally from acetate it goes to methane and carbon dioxide that by the process called methanogenesis. Okay, so formation of methane is methanogenesis. Formation of acetate, so acetogenesis. Okay, and fermentation is a anoxic process. I have explained this in the previous sessions also. So in the absence of oxygen, the respiration process is basically fermentation. Okay, and breakdown of polymers into monomers is hydrolysis. Is it clear now? Everybody understood this? Okay. Then I think I can move on to the next question. Question number 8. Methanogens belong to which of the following domains? So we just learnt about methanogenesis, and it is carried out by a group of bacteria called methanogens. Now you have to tell they belong to which of the following domains. Option A, fungus. Option B, eukarya. Option C, archaea. And option D, bacteria. So I can tell you that I've already told you that these are bacteria. So among bacteria, we have two groups, two domains, eukarya and archaea. So methanogens belong to one of these two domains. So this was your hint. Now tell me what would be the correct answer. Write your answers in the chat box. Okay, I have one answer, option C, that is archaea. What about others? Anybody else? Anybody else knows the answer to this question? Write the answers in the chat box. I'm getting only one answer. Answers from one person only. What about others? Others in the call, can you hear me? Can you see my screen? If yes, then write the answers in the chat box. Okay, let's see the correct answer. The correct answer is option C, archaea. So you are right. So methanogens can produce methane during metabolism, as already I have discussed. So they belong to the archaea domain of bacteria. Okay. So I had shown you here, methanogens are basically the ones that are converting the acetates into methane by the process called methanogenesis. Okay. And they belong to archaea domain. Is this clear? If anybody has any doubt, please ask. Okay, if there's no doubt, then I can move on to the next question. What is the recommended period of time in days for a BOD test? What is the recommended period of time in days for a BOD test? Option A 1 day, option B 5 days, option C 2 days, option D 7 days. Recommended period of time for a BOD test. I have one answer in the chat box, option B, that is 5 days. Okay. So BOD is biological oxygen demand. So it is a test done for the wastewater to determine if the wastewater treatment is done properly or not. Okay. So what is the recommended period of time for BOD test? I have one answer, option B. What about others? Everybody write your answers in the chat box. Okay, let's see the correct answer. The correct answer is option B. So you are right. Recommended period of time for the standard BOD test is 5 days according to APHA, AWWA, and WEF. So APHA is American Public Health Association. AWWA is I think American Wastewater uh, Association or something, and then you have WEF, that is Wastewater Environment Federation. Okay, so or Water Environment Federation. So basically, these are government organization, and they have created these guidelines. So according to the guidelines, it is recommended to test the BOD after 5 days. Okay, that's clear to everyone. Moving on to the next question, question number 10. Dash is the largest source of sulfur dioxide, that is SO2, in the atmosphere. Dash is the largest source of SO2 in the atmosphere. Option A, burning of fossil fuels. Option B, locomotives, ships, and vehicles. Option C, volcanoes. And option D, extraction of metals from ores. Burning of fossil fuels, locomotives, ships, and vehicles, volcanoes, and extraction of metals from ores. What is the largest source of sulfur dioxide in the atmosphere? Write your answers in the chat box. I have one answer in the chat box, option A. That is burning of fossil fuel. Okay. Anybody else wants to answer? Largest source of sulfur dioxide in the atmosphere. What would be the answer? Write it on the chat box. Okay, let's see the correct answer. The correct answer is option A, burning of fossil fuels. So you are right. So largest source of sulfur dioxide in the atmosphere is burning of fossil fuels. Okay. Moving on to the next question now. Question number 11. Sulfur dioxide released in the atmosphere is converted into dash and dash. SO2 that is released into the atmosphere, it is converted into dash and dash. Option A, sulfur and oxygen. Option B, sulfuric acids and sulfates. Option C, hydrogen sulfide and oxygen. And option D, sulfonic acid and oxygen. Write your answers in the chat box. Sulfur dioxide released into the atmosphere is converted into dash and dash. Option A is sulfur and oxygen. Option B is sulfuric acid and sulfates. Option C is hydrogen sulfide and oxygen. And option D, sulfonic acid and oxygen. I have one answer in the chat box, says option A, that is sulfur and oxygen. What about others? Anyone else wants to answer this question? Sulfur dioxide into the atmosphere is converted into option A, sulfur and oxygen, option B, sulfuric acid and sulfates, option C, hydrogen sulfide and oxygen, and option D, sulfonic acid and oxygen. Anyone else wants to answer? Write your answers in the chat box. Okay, I have one more answer. Option B. Okay, let's see the correct answer. The correct answer is option B. So you're right. Sulfuric acids and sulfates. So sulfur dioxide is basically into that is released into the atmosphere. It is converted into sulfuric acids and sulfates. So they are basically oxidized. So sulfur dioxide, the oxidation state of sulfur is +4, right? So sulfuric acids or sulfates, in that case, the oxidation state of sulfur becomes +6. So sulfur is basically oxidized. Sulfur dioxide is oxidized into sulfuric acid and sulfates. And you must have heard about acid rains, right? So this is the reason we are having acid rains. So all the sulfur dioxide that is released into the atmosphere, and we have seen why what is the source of sulfur dioxide. The source was burning of fossil fuels. So all the fossil fuels that are burning, they are causing sulfur dioxide release into the atmosphere. And when once it reaches the atmosphere, we have plenty of oxygen to oxidize sulfur dioxide further into sulfuric acids. And when it rains, these acids basically pour and reach the ground along with the rainwater. Okay, so this is how we get acid rain. So sulfur dioxide is converted into sulfuric acid and sulfates in the atmosphere. It is basically oxidized. Is that clear? Anybody has any doubt? Okay, moving on to the next question. Question number 12. Biogas generated at the end of the anaerobic digestion is mainly composed of dash and dash. Biogas generated at the end of the anaerobic digestion is mainly composed of option A, ammonia and methane, option B, H2S and carbon dioxide, option C, CH4, that is methane and carbon dioxide, option D, methane and carbon monoxide. Biogas generated at the end of the anaerobic digestion is mainly composed of dash and dash. Option A, ammonia and methane. Option B, H2S and carbon dioxide. Option C, methane and carbon dioxide. And option D, methane and carbon monoxide. Write your answers in the chat box. Okay, if Smita is confused between C and D. Okay. I have one more answer here, option C. What about others? Everybody write the answers. Okay, I have one more answer in the chat box, says option C. So let's see the correct answer. Option C is the correct answer. So you both are right. Uh, so anaerobic digestion of biogas involves microorganisms breaking down the organic matter into methane and carbon dioxide. So remember I told you about methanogenesis. This is where that process is happening, and final uh, methane and carbon dioxide is produced. So this is nothing but biogas. Okay. So organic matter, that is carbohydrate, proteins, these are polymers. They are broken down into monomers by hydrolysis, then converted into acetate, and that acetate is converted into methane and carbon dioxide, that is biogas, by the process called methanogenesis and by the organism called methanogens. Okay. And remember, methanogens belong to which domain? They belong to archaea domain. Okay. So these are the bacteria, archaebacteria basically. That's clear to everybody? Is this clear to everybody? Hmm. Okay. Moving on to the next question, question number 13. Which enzyme can catabolize starch? Which enzyme can catabolize starch? Option A, cellulase. Option B, dextranase. Option C, chitinase. And option D, amylase. Which enzyme can catabolize starch? Catabolize means break down. So which enzyme can break down the starch molecule? What can break starch? Cellulase, dextranase, chitinase, or amylase? Write the answers in the chat box. Okay, I have two answers in the chat box. Both of them are option D. What about others? Anyone else wants to answer? Anyone else wants to answer? Come on, write the answers. What can catabolize starch? Cellulase, dextranase, chitinase, amylase. Two people think it's amylase. What about others? Okay, let's see the answer. The correct answer is amylase. So both of you are right. So amylase catalyzes the hydrolysis of starch into simple sugars. So starch is a straight chain polymer of glucose with alpha 1,4 glycosidic bonds between individual D-glucose molecules. So we have D-glucose molecules connected together with alpha 1,4 linkages. So it's a straight chain polymer and it is broken down by amylase. So it basically breaks down the alpha 1,4 linkage and individual glucose molecules are released. Okay. So starch, a straight chain polymer of glucose with alpha 1,4 linkages, alpha 1,4 glycosidic linkages. These are broken down. These linkages are broken down by amylase to form individual glucose molecules. Okay. So starch is basically a carbohydrate. So I told you in the previous this we had complex polymers like carbohydrates. So starch would be a carbohydrate which is hydrolyzed. So this is the process hydrolysis. And what enzyme is doing this hydrolysis process? Amylase is doing this hydrolysis process. And what is the monomer we are getting? We are getting the monomer as sugar. What is the sugar here? Glucose is the sugar here. Okay. So are you able to relate everything now? Is this clear? Everybody understood? Moving on to the next question then. Question number 14. Fatty acids are metabolized through which of the following process? Option A, alpha oxidation pathway. Option B, beta oxidation pathway. Option C, hydrolysis reaction. Option D, oxidative phosphorylation. Fatty acids are metabolized through which of the following process? Alpha oxidation pathway, beta oxidation pathway, hydrolysis reaction, and oxidative phosphorylation. Write the answers in the chat box. Fatty acid metabolism. What is the process of fatty acid metabolism? Alpha oxidation, beta oxidation, hydrolysis, or oxidative phosphorylation? I have one answer in the chat box, option B, that is beta oxidation. What about others? Anyone else wants to answer? Okay, let's see the correct answer. The correct answer is option B. So you are right. Option B, fatty acid, that is beta oxidation pathway. So fatty acids are metabolized through this pathway. So what is beta oxidation? Basically, fatty acids are long aliphatic hydrocarbon chain with uh, carboxylic acid group at the end. Okay. So, if this is the carboxylic acid group, so this would be the alpha carbon and this would be the beta carbon. So, always after the beta carbon, the cut occurs. So, the molecule that is released from the fatty acid, long chain fatty acid, is acetyl coenzyme A that can enter the TCA cycle, citric acid cycle, that I've already explained in one of the previous sessions. And we have the remaining chain. So if let's say how many carbon it has? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and this is 14. So 14 carbons are there. So after the beta carbon, the cut will happen. So this will be removed as acetyl coenzyme A. and this carbon will get oxidized into the carboxylic acid group. So this will have the coenzyme A attached. And now you will have, so out of 14, 2 carbons are removed. So you will have now 12 carbons. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So like this, after every step, so after this step, only 10 carbon will be remaining. Then 8 will be remaining. Then 6 will be remaining. 4 will be remaining. Then finally, we'll have acetyl coenzyme A removed. So uh, by this beta oxidation process, repeatedly, 2 carbons are removed in the form of acetyl coenzyme A, and all these acetyl coenzyme A, they keep on entering the citric acid cycle. Okay. So this is how fatty acids are metabolized inside the cell. Okay. So you remember glucose was metabolized inside the cell by the process called glycolysis. So glucose was converted into pyruvate, and then pyruvate was converted into acetyl coenzyme, which was entering the citric acid cycle. Similarly, fatty acids are also metabolized in our cells. So how fats are metabolized in our cells? It is metabolized by the process called beta oxidation. Okay. Is this clear to everybody? If anybody has any doubt in this process, let me know. Everybody understood what beta oxidation is, how fats are metabolized inside the cell? Is this clear to everyone? Anybody has any doubt? Okay. Moving on to the next question then. Question number 15. Is the following sentence true or false? The sentence is fatty acid metabolism is an ATP utilizing process. Is it true or false? You just saw the fatty acid metabolism by beta oxidation. Does it utilize ATP? True or false? Tell me. Write your answers in the chat box. Is it option A or option B? Okay, I have one answer that says option A. That is, it is true that fatty acid metabolism is an ATP utilizing process. Okay. What about others? Anyone else wants to answer? Do you think ATP is utilized in fatty acid metabolism, or it is produced during fatty acid metabolism? If it is utilized, then option A would be correct. If it is produced, then option B would be correct. Tell me the correct answer. Anyone else? I have only one answer in the chat box. What about others? Where are you? Write your answers in the chat box, everyone. Okay, let's see the answer. The correct answer is actually false. The statement is false. In during the fatty acid metabolism, during beta oxidation, ATP is actually generated. So here you can see in this whole process, every time acetyl coenzyme is removed from the fatty acid chain, ATP molecules are generated. Okay, so in a beta oxidation process, ATP is generated, it is not utilized. So the statement fatty acid metabolism is an ATP utilizing process, it is false. Okay, is this clear to everyone? Everybody understood? ATP is generated in the fatty acid metabolic process. Okay, ATP is generated during beta oxidation. I hope that's clear to everybody, right? Okay, moving on to the next question. The range of total Geldal nitrogen in sewage is normally around option A 10 to 15 mg per liter, option B 20 to 80 mg per liter, option C 30 to 50 mg per liter, and option D 5 to 20 mg per liter. The range of total Geldal nitrogen in sewage is normally around option A 10 to 15 mg per liter, option B 20 to 80 mg per liter, option C 30 to 50 mg per liter, and option D 5 to 20 mg per liter. Write your answers on the chat box. What is the range of total Geldal nitrogen in sewage? Anyone knows? Okay, I'll tell you the answer to this, since this is just knowledge base. So option B is the correct answer. 20 to 80 mg per liter is the total range of total Geldal nitrogen in sewage. Okay. So option B is the correct answer. Moving on to the next question. According to the new National Green Tribunal guideline, what is the maximum BOD value at 27 degrees Celsius for treated wastewater being discharged into any inland surface water body? So I told you BOD is the biological oxygen demand. It is the measure that we do to check the wastewater treatment is being done properly or not. So, according to the new National Green Tribunal guidelines, what is the maximum BOD level for the treated wastewater being discharged into any inland surface water body? Option A is 80 mg per liter, option B 30 mg per liter, option C 10 mg per liter, and option D 5 mg per liter. What would be the correct answer? Anybody? If you know the answer, please write it in the chat box. This is given in your lecture videos. So if you have watched the lecture videos, you should be able to answer this. I have one answer, option B, that is 30 mg per liter. What about others? Anyone else wants to answer? So let's see the correct answer. The correct answer is option C, 10 mg per liter. So 10 mg per liter is the maximum BOD value for treated wastewater being discharged into any inland surface water body. So 10 mg per liter is the revised sewage discharge standard for wastewater being discharged in mega and metropolitan cities. So in India, mega cities are defined as the cities with population greater than 10 million, and metropolitan cities are defined as population greater than 1 million. So you can see here the revised guidelines given. So in the mega and metropolitan cities, the pH of the treated water should be between 5.5 to 9, and the BOD should be 10 mg per liter. In class 1 cities, the pH range is same. BOD can be 20, and in other cities, it can be up to 30. And in deep marine outfalls, it can for that also the BOD can be 30. Okay. Moving on to the next question now. Question number 18th. Ruminant digestive system is analogous to. The ruminant digestive system is analogous to option A, aerobic digestion tank, option B, anaerobic digestion tank, option C, trickling bed reactor, and option D, continuous stir tank reactor. Ruminant digestive system is analogous to option A, aerobic digestion tank, option B, anaerobic digestion tank, option C, trickling bed reactor, option D, continuous stir tank reactor. Write your answers in the chat box. Everyone write your answers in the chat box. The ruminant digestive system is analogous to aerobic digestion tank, anaerobic digestion tank, trickling bed reactor, or continuous stir tank reactor. What would be the correct answer? Anyone? I have one answer in the chat box says option A. That is aerobic digestion tank. Anyone else? Okay, another answer in the chat box is option B. That is anaerobic digestion tank. Anyone else? Is there anybody who agrees with option A or option B? Okay, let's see the correct answer. The correct answer is option B, anaerobic digestion tank. So, Neelma, you were right. Uh, so, ruminant digestive system, ruminant basically are the animals like cow. or buffalo. So they have a different kind of digestive system, and their digestive system acts exactly like an anaerobic digestion tank. So microbes digest and ferment the plant material. So cows eat plant material. They eat uh, raw grass and all, right? So dried grass and everything. So all those plant materials, those are basically how do they digest those complex plant materials. So they basically have microbes in their gut, and they digest and ferment those plant material to produce to provide energy. And in the process, gases like carbon dioxide and methane are released. So remember, we talked about methanogenesis and we talked about biogas. So basically, biogas is produced uh, inside these cattle's stomach. So it's the same process that happens in the anaerobic digestion tank. So in the wastewater treatment plant and the sewage treatment plant, we have an anaerobic digestion tank where also microbes are allowed to ferment the organic matter to produce carbon dioxide and methane under anaerobic conditions. So same thing is happening in the digestive system of these cattles. Okay, is this clear? So basically, this is biogas, carbon dioxide and methane. This is the biogas, and this is produced by the process of methanogenesis. Is this clear? Okay, let's move on to the next question. Question number 19. Which of the following sources is responsible for the most amount of methane emitted globally? Which of the following sources is responsible for the most amount of methane emitted globally? Option A, coal mining. Option B, rice cultivation. Option C, wastewater. And option D, enteric fermentation. Which of the following sources is responsible for the most amount of methane emitted globally? Coal mining, rice cultivation, wastewater, and enteric fermentation. What would be the correct answer? Write your answers in the chat box. Write your answers in the chat box. I have one answer in the chat box, enteric fermentation. What about others? Anyone else wants to answer? I have one more answer, option A, that is coal mining. Anyone else? Anyone else wants to answer? What do you think is the major source of methane emitted globally? Okay, let's see the correct answer. The correct answer is option D, enteric fermentation. So enteric fermentation is actually the major source of methane production. So enteric fermentation is a digestive process of the ruminant animals that produces methane as a byproduct, as I explained in the previous question. So the digestive system of the ruminant animals basically is similar to the anaerobic digestion tank where biogas methane is produced, and enteric fermentation is actually the major contributor of this uh, methane gas emission, and it is actually a major cause of global warming. So methane is major one of the major causes of global warming. So the gas produced by the livestock, by the ruminant animals, this is the major source of the methane emitted globally. Okay, so enteric fermentation is a part of digestive process in the ruminant animals, as I've told, where microbes in the digestive tract decompose and ferment the food, producing methane as byproduct. All the other activities do not contribute to methane production as much as enteric fermentation. Okay. So these gases are released basically by the excretion process of the uh, of these animals. So you know that cow dung is used as a biofuel. People in earlier days used to use cow dung as a source of fuel because it is rich in methane and biogas. Okay, so it is very combustible and it is a good source of fuel. It, but obviously, since it releases methane, it causes a lot of environmental problem. It is the major source of global warming, and you all know the repercussions of that. So, any doubt? Is it clear to everybody? If you have any doubt in any of the questions discussed till now, please ask. Moving on to the next question. Which pair of enzymes catalyze oxidation of hydrocarbons under aerobic conditions? Which pair of enzymes catalyze the oxidation of hydrocarbons under aerobic conditions? Option A, lipase and amylase. Option B, cellulase and hydrogenase. Option C, monooxygenase and dioxygenase. And option D, ligase and lipase. Which pair of enzymes catalyze the oxidation of hydrocarbons under aerobic conditions? Lipase and amylase, cellulase and hydrogenase, monooxygenase and dioxygenase, and ligase and lipase. Which of the following carries out the oxidation of hydrocarbons under aerobic conditions? Write your answers in the chat box. What would be the correct answer? Okay, I have one answer in the chat box. C, option C, that is monooxygenase and dioxygenase. What about others? Everybody write your answers. What do you think is the correct answer? What is the correct answer, right? Anyone else wants to answer? I'm sorry. There are some people who are trying to join the call at 8:40. The class has started from 7. I'm not going to allow anyone to enter the class just 15 minutes when the class is remaining. Okay, let's see the correct answer. The correct answer is option C. So you are right. Monooxygenase and dioxygenases are two enzymes that help in the catalysis of hydrocarbon oxidation under aerobic condition. So basically, just a second. Yeah. So here you can see the oxidation of aliphatic carbons is shown. So in case of aliphatic hydrocarbons, uh, it is first oxidized by a monooxygenase into an alcohol. Then it is further oxidized into aldehyde and then carboxylic acid. So once the carboxylic acid is forming for the aliphatic hydrocarbon, it is nothing but a fatty acid. And then, by beta oxidation, fatty acids are converted into acetyl coenzyme. Okay, so monooxygenase, they can convert the hydrocarbons into octanol, and then further it can convert into aldehyde. So in this case, octanol, and then octanoic acid, that is the carboxylic acid, and that carboxylic acid is basically the fatty acid, and it is metabolized by beta oxidation. In case of aromatic hydrocarbons like benzene, so in case of benzene, benzene monooxygenase can form an epoxide, which can further be converted into catechol and metabolized. For catechols, there are dioxygenases, which can basically metabolize the catechol, break down the aromatic chain. Then for toluenes, we have toluene dioxygenase. So these are just examples of different aromatic compounds and how they are metabolized. Okay. So basically, hydrocarbons, aromatic or aliphatic, they are metabolized by monooxygenases and dioxygenases. Okay. Moving on to the next question. Question number 21. During lightning, the high temperatures and pressures created in the air converts nitrogen into option A chloroform, option B ammonia, option C oxides of nitrogen, and option D oxygen. During lightning, the high temperature and pressure created in the air converts nitrogen into dash. Write your answers in the chat box. What would be the correct answer? During lightning, the high temperature and pressure created in the air converts nitrogen into option A chloroform, option B ammonia, option C oxides of nitrogen, and option D oxygen. Anybody knows the answer? Okay, I have one answer in the chat box. Option C, that is oxides of nitrogen. What about others? Anyone else wants to answer? Anyone else? Okay, let's see the answer. The correct answer is option C, oxides of nitrogen. So during the nitrogen cycle, I've already told you the different processes that are involved in the nitrogen cycle. So in atmospheric nitrogen, it is either fixed by the soil bacteria and converted into ammonia, or by lightning, this atmospheric nitrogen can be converted into oxides of nitrogen, that is nitrites and nitrates directly. Okay, so by physical process during lightning, the high temperature and pressure are created in the air, which converts nitrogen into oxides of nitrogen and dissolves in the water and comes down along with the rain. This is called nitrification. So direct conversion of nitrogen to nitrates, this is nitrification. Another process is nitrogen fixation, where nitrogen is converted into ammonia. That ammonia is also converted into nitrites and nitrates. That is also nitrification process. Then these nitrates are converted back to atmospheric nitrogen by the process of denitrification. Okay. So you have nitrogen fixation, you have ammonification, you have nitrification, and then you have denitrification, that is going back to the atmosphere. So this is how the cycle keeps on going. Is this clear? Is this clear to everybody? Moving on to the next question then. Which of the following is a sedimentary type of biogeochemical cycle? Option A, phosphorus and carbon monoxide. Option B, oxygen and nitrogen. Option C, phosphate and nitrogen. Option D, phosphate and sulfur. Which of the following is a sedimentary type of biogeochemical cycle? Write your answers in the chat box. What would be the correct answer? Which of the following is sedimentary type of biogeochemical cycle? Phosphorus and carbon monoxide, oxygen and nitrogen, phosphorus and phosphate and nitrogen, and phosphate and sulfur. I have one answer in the chat box, option D. Okay, that is phosphate and sulfur. Anyone else wants to answer? So what do we mean by sedimentary type of biogeochemical cycle? It basically means uh, it's a, like in case of nitrogen cycle, the nitrogen is cycled from the atmosphere to the soil and back to the atmosphere, right? Similarly, in these sedimentary biogeochemical cycles, the elements are moved from the earth's crust or water bodies back to the sediments of uh, earth, okay, back to the sediment. Okay, so there is no involvement of atmospheric air. So now you can see the correct answer would be option D, phosphate and sulfur, because all the other three options that we have, all of them have gases in them. So they will go back to the atmosphere. But in case of phosphate and sulfur, they will go back to the sediment. They will go back to the earth's crust. Okay. So earth's crust is the reservoir for these cycles. Okay. So carbon monoxide, oxygen, nitrogen, all of these are gases. So they are going to go back to the atmosphere, but phosphorus and sulfur, they will go back to the Earth's crust. Okay. So they move from the they are moved from the water bodies and other areas to the uh, sediment. Okay. Is this clear to everyone? Okay. Next question is aerobic oxidation of H2S is carried out by which of the following bacteria? Aerobic oxidation of H2S is carried out by which of the following bacteria? Option A, purple sulfur bacteria. Option B, green sulfur bacteria. Option C, Thiobacillus and Veggiatoa. And option D, desulfovibrio and desulfobacter. Aerobic oxidation of hydrogen sulfide is carried out by which of the following bacteria? Option A, purple sulfur bacteria. Option B, green sulfur bacteria. Option C, Thiobacillus and Veggiatoa. And option D, desulfovibrio and desulfobacter. Write your answers in the chat box. What would be the correct answer to this? Which organism carries out the aerobic oxidation of hydrogen sulfide? So this is a part of sulfur cycle. Come on, write your answers. Even if you don't know, take a guess. Okay, let's see the correct answer. The correct answer is Thiobacillus and Veggiatoa, option C. So this is the sulfur cycle. So sulfur cycle goes on in two parts. So aerobic part and the anaerobic part. So you can see it is divided into two different halves. So in the aerobic part, the elemental sulfur, it is either oxidized to sulfates, and that those sulfates, these are then assimilated to the SH groups of the protein, or those sulfates are then reduced to hydrogen sulfide by bacteria like desulfovibrio or desulfobacter. These hydrogen sulfides are again oxidized to elemental sulfur by Thiobacillus and Veggiatoa. Okay. So you can see here reduction is happening. So similar to nitrogen cycle where nitrates were reduced to nitrogen, here sulfates are reduced to sulfur by the intermediate formation of hydrogen sulfide. These hydrogen sulfides are then oxidized to sulfur by uh, Thiobacillus and Veggiatoa in the aerobic case. In case of anaerobic uh, process, elemental sulfur is oxidized into sulfates by purple and green phototrophic bacteria. These sulfates are assimilated into the proteins and then converted into hydrogen sulfides, and hydrogen sulfides are converted back to the elemental sulfur by purple and green sulfur bacteria by, by anaerobic process. Okay, so aerobically, hydrogen sulfides are oxidized to sulfur by Thiobacillus and Veggiatoa. Anaerobically, hydrogen sulfides are oxidized to sulfur by purple and green sulfur bacteria. Okay, and desulfovibrio and desulfobacter, they basically do the reduction process. So The other two processes are the other processes, oxidative process. This is a reduction process where the sulfates are reduced to hydrogen sulfide, and that is carried out by desulfovibrio and desulfobacter. So you had purple sulfobacteria, green sulfobacteria, thiobacillus and Beggiatoa, and desulfovibrio and desulfobacter, right? So desulfovibrio and desulfobacter, these bacteria are carrying out the reduction process of sulfate to hydrogen sulfide. Thiobacillus and Beggiatoa carries out the oxidation of hydrogen sulfide to sulfur aerobically, and these two carry out this process anaerobically. Okay, is this clear to everybody? This was the last question of the session. I hope everything is clear. If you have any doubt, please let me know. So today was the last problem solving session. So tomorrow we'll have one extra session where I'll be discussing all the 12 uh, lectures that have been, like 12-week lectures that have been discussed till now. So we'll discuss different topics. I'll prepare some questions related to each week's topic. And I would request all of you to join that session. And if you have any doubt regarding any of the session that we had previously, so we till including today's session, we had 12 sessions, right? So out of those 12 sessions, if you have any doubt in any of the topics discussed, let me know tomorrow, and I'll if you have any doubt, you can ask me now also so that I can prepare for it tomorrow. Otherwise, let me know tomorrow and I'll try to clarify the doubt. So tomorrow will be the last session for this course. After that, you will have your exams. So prepare well for your exams. Anybody has any doubt or anything you want to ask? Everything is clear then, I hope. Okay, if there's no doubt or no questions, then I think I can end this session today. I'll see you guys tomorrow. So tomorrow will be the last session. So be prepared with your questions if you have any. Um, I'll try to clarify your doubts. Okay, so thank you all for joining. I'm ending this session now. Bye.